I can see what I'm talking about. And then let's see, Hercules is the little square with the blue marks. Is that right? And Argus is behind it with the red marks, or do I have that off?
Other way around, yeah. So Argus is the blue one closer to the ship, and Hercules is stretched out south,
okay.
marking a red line.
And Hercules will be the video that we'll be viewing, will be the primary video that we'll be viewing the seafloor with. But Argus plays a really important role too. And maybe Jake can explain the role that Argus plays in this operation when you have a chance.
Sure, yeah. It's probably got two primary roles. One is removing the surface motion from, or decoupling it from Hercules, the ship motion. So that's how you get that really clear video, really not bouncing around, and those good shots. And then the second feature that it provides is kind of this extra spatial view for the Hercules pilot to see their surroundings and maneuver around. And so that the HD camera, the Zeus, is actually the same HD camera that's on Hercules, that's on Argus. And so you get that high quality imagery of kind of your surroundings, and it, I don't know, gives you a better perspective of, or the Herc pilot a better perspective of where they're going and where they're moving. So those are probably the two primary functions of Argus. But it also provides an extra data point for different sensors of such as depth, and right now it's got the sub bottom going, so it's found the bottom, the first sensor to find the bottom. So we're like 70 meters away from the seafloor, and yeah, there's a whole suite of sensors on Argus, similar to what we have on Hercules. So it can run also as a standalone system and do stuff like side scan sonar and mapping.
70 meters, holy cow, I better slow down.
Yeah. And you're 16 meters below me, so.
Yeah, you're 40 off the deck. I just switched you over to DVL.
Roger. I'll stop it when Argus is 50, and I'll fly under you towards the hill.
Roger.
And so are both ROVs equipped with like a CTD? I know other sensors as well, altimeter, and but is there a CTD mounted? I'm sure there must be.
There's a CTD on Hercules, but both ROVs have sonar, depth, altitude, lights, cameras, compass, thrusters.
You can see both sonars on satellite feed 3 or channel 3.
I'll switch over to that.
What's going to be our bearing, Rennie?
Yep, just checking that. Right.
Oh, yeah.
Roughly northeast, isn't it?
On the left is
roughly northeast.
Hercules sonar.
040.
On the right is Argus's sonar.
You can light up your thrusters if you want, and hold heading at 045. Only the aft one, yeah.
It's already fired up.
Roger.
And Argus's sonar,
under you, 20 meters.
You can see a target behind it, and I believe that is Hercules.
To the south. Yeah. And is it 130, is the, let's see, the radius 139 meters of that sonar, or what's the distance that it's?
I think on the left, Hercules is 10-meter intervals on those lines. And on the right, on Argus, it's 20?
Yes.
Yep.
Correct.
Okay.
And it's not necessarily south. I think that when you're looking up, that's the heading of the ROVs. So that's always forward for the ROVs and aft of the ROVs.
Okay. Oh, I do see the intervals in the upper left corner of the screen. Oh, that's cool. Thanks for showing that.
There you go, visual in Argus HD.
What's that?
Visual in Argus HD.
Yeah, right. Okay. I can continue down.
Roger.
Hey, what happened there?
Part of it, I think, is the thruster can't keep up. The one thruster.
I'm pulling on it.
Yeah.
And come on, stop there for a second, Jake.
I'm stopped.
Can you change that to 20 for me?
Roger.
It gets me every time. Thanks.
Just see you in down cam. Yeah, there you are.
Yeah, sorry, I was not looking at the nav screen on the wrong resolution. Well, I didn't notice it was set to 50.
Coming back down.
Roger. $10 meters.
10 meters high. Logging on bottom.
You can stop when you get to 20 meters. Yeah.
Roger.
And here we are on the bottom.
Well, we did touch there. Auto altitude, the first time you use it, likes to find out exactly where the bottom is.
It's ground truthing.
How high up are you off the bottom before the DVL detects the bottom?
About 80 meters nowadays. Seems to be working.
Oh, okay.
The fourth beam kind of kicks in at least around 70.
So you got plenty of warning
Yeah.
if it's murky or something.
There's a bug in the system that the first time you engage auto altitude, it drives the thrusters, drives the vehicle down hard.
Mm.
And so you got to turn it on, turn it off, and then turn it back on. And I thought I did that, but usually do that about 5 meters. Sometimes it takes a couple cycles of it. And I tried it at 2 meters, which is fatal.
I'm gonna wait for this dust for 1 second.
Oh boy.
I'll come up. I thought that dust is a reality, white balanced. All right, it's clear water now, Steve.
Sure. All right, we're gonna go black for a second.
Roger. You can come down another 5 if you want,
Hello,
Jake.
this is Lisa. I'm not hearing anything.
Hi,
Hear
Lisa.
us?
Hi,
Okay,
Lisa.
and white balancing.
I
Okay,
can't hear you guys. So, Steve, I hope you can do your magic. Or,
check, check. You can't hear us?
I let her know in the chat that we can hear her, Steve.
okay, I can hear you now. It was just,
Great.
thanks.
Hi, Lisa.
Oh, good.
Hey,
Hi,
good
Lisa.
afternoon, everyone. Hi, hi.
We
I
just
just got off.
landed.
Yeah, I saw. Did you see the billfish?
Is that what that was?
We saw a big fish. We didn't know what it was.
Yeah, I saw
Yeah,
it.
No,
it
how
was,
did I miss it?
it was at, so I wrote down the time, 12:33 PST
12:33.
at 450 meters. It was some type of billfish. It looked like, a sword. It wasn't just swordfish. It had a wider bill.
Bill?
Yeah, but it was definitely fast swimming, high speed type shape
Hmm.
fish.
Wow.
It was pretty cool. I was like, wow, did you see that?
Have to rewind the video after it's been archived.
Oh, so Dan Giger is Daniel. He's agreeing from Santa Barbara
What
Yeah,
time?
I
Museum of
think
Natural
I
History.
might have stepped away for a
Wh-
moment.
What time was
See
the
if this
swordfish?
goes to show.
12:33.
At 12:33 PST, so somewhere around there. I'm going. I didn't get the UTC time, so we can always go
UTC
back.
6 or 7 hours different now. I can't.
Yeah,
I'm gonna
7
go through
hours.
time.
All right, so are we
It's
all
7
sorted
hours.
out down here?
Yep.
All right, we're ready to go upslope as long as that's good, unless there's any tasks you want to complete right here.
Dan Geiger is with us and pointed out a snail.
Hey.
Dan, do you want that?
Can you put the braid arm all the way to the left?
I think I was covering up my mic. Let's see if he responds.
UTC time right now is
He has some already.
19.
Oh, sorry, what was that?
I said UTC time right now is 1957.
So it was at 1933 then, right, Lisa? The billfish swam by.
Say that again. I'm sorry, Di.
If it's
12:33.
1950, yeah,
Yes,
which is
then it would have been
1933 UTC.
yes, something around there. Yeah. And Taylor Ann is confirming. Thanks, Taylor Ann.
That's shoulder and
No
wrist all
problem.
the way left.
Right, that's better, thanks.
So Dan says he's got some already, and we can proceed then.
Proceed, Roger. Okay, we'll go start tracking up slope 040.
Roger.
And so I was just on the 12:30 call, and I spoke with Lizzie, who's the author of this dive plan, and she said between EA and ECED, what we're basically doing is we're verifying the backscatter. So we're ground truthing it, and we can move fairly quickly through here. We can do transects at a faster speed, whatever speed Dan or Jake feel comfortable with. And when we get to the photo mosaics, we had a very good discussion about what we're trying to do here with the photo mosaics. And they're very happy with what we did on the dive previously, the 3 hours that we spent on the Acanthogorgia area, the 10 by 10 meters that we did. But if we do another one, we should just pick a boulder outcrop with some interesting features and do it more like a 3D mosaic, like a whale fall.
Roger. So
Okay.
and then so that'll be for the 3D photo mosaic. And then is there any plans for the 2D habitat mapping or no?
The 2D habitat mapping that we did last night, that'll be it for now.
Okay, Roger. Copy.
Yeah.
Okay. Okay,
Thanks.
so that's clear.
And what else did she say? I'm just looking at my notes here. So backscatter validation, we can do faster transects. And when we get to ED, which I don't think we will, we have many choices. It's like Goldilocks. Do we want it hot? Do we want it cold? Do we want it medium? So there's different routes that we can take. But I don't think we'll get there. We'll probably have
On
those
this shift.
decisions to make on our next shift.
All right, well then I don't think the slope is going to be too significant. So if we want to head up at 0.5.
Sure.
Bridge nav.
Yeah, and if we want to start a transect, we could do that.
Let's increase speed to 0.5.
Yeah, can do.
Yeah.
Transect out of the gate.
We can do it. Yeah, if you guys are okay with that, at that speed, it would be fine.
Sure.
0.5
as soon as
for
Argus
a
starts
transect
moving,
on this.
we'll go ahead.
Yeah. Okay.
Thank you.
Thank you.
Did you all do your introductions already?
We did, yeah.
We did,
Okay,
so
well, then
we're just waiting for you.
Hi. Hi, everybody. This is Lisa Vonink. I work for the Office of National Marine Sanctuaries in their West Coast Regional Office. I'm their policy coordinator, and I have the great privilege of working with the 5 West Coast sanctuaries, of which the Nautilus has done many cooperative cruises with and explorations for deep sea corals, not just this year, but in previous years as well. And we are right now in Channel Islands National Marine Sanctuary. We're at the Footprint Marine Reserve.
Yeah, and we're sort of across I like to call it sort of a valley. We're kind of at the base, the northern side of Piggy Bank, and we're going to be crossing across toward the Footprint Bank. But yes, we are within the Footprint reserve.
Okay, that's going to be start of transect.
Roger that. Start a transect.
Okay.
1837.
I'm going to mute so I can type.
Are those little thorny heads with the?
They're all on.
Big
Yes,
pectorals.
and
Yeah,
thorny heads, and I
long
think
spine.
I saw long spine.
A longer
Yep.
Yeah, I saw the same thing.
Yeah, as they erect their dorsal spines, you can see that one is longer, noticeably longer than the others. And at this depth, that would make sense that they're long spine thorny heads.
Yep.
I'm going to type in the time of this transect start, so I'm going to mute while I'm typing.
I'm wearing my baseball cap backwards. My ears thank you for that tip. It feels so much better. So for those of you who are listening in, what I'm talking about is I have a new headset, the kind that looks like Princess Leia, the big ear muffs, and it really hurts when because I wear glasses, and the Nautilus crew told me what I could do, wear a baseball cap first, so that helps. It cushions it. Seeing different kinds of sea stars, echinoderms, bat stars, more thorny heads, different sizes. There's a sponge up there. Ooh, a flatfish. That is Tom, help me out. Not a petrale. It has interesting patterns.
A Dover sole.
Dover sole. Are they the ones with the fin that always sticks up?
No, that's a wreck. I think that was a Dover sole. We could also see deep sea sole down at this depth as well.
interesting sponges when we hit some hard bottom
We are
features.
I think that's another Dover sole over to the left.
And that was a grenadier, I think, in the water column.
That's a little, yeah, a little grenadier.
So that's another Rexol dye.
Coming up here,
Well, we just
think
passed
that's
it.
another. Oh, okay. This one
Oh,
that we're just going over now is a Dover.
sorry. You're right. Dover sole. Okay, got to write this down.
That little fish that just dive bombed, I'm losing the name, but it'll pop into, I won't shout it out when it pops into mine. It's an eel pout. It's a type of eel pout, I think. We're getting a few rocks here. Nice sponge with a thorny, it looks like a long fine thorny head beside it, displaying that. That's actually the third dorsal spine. The first two can be hard to see. And that fish is roughly 25 centimeters, I'd say. These two green dots in the middle of the screen are lasers, and they're spaced 10 centimeters apart, which is about just under 4 inches, and they help us estimate the size of things. And so we're doing a quantitative survey transect right now that will go for about 200 meters at least. And video analysts will review this video and record the habitat along the way and the critters, the fishes, coral sponges, and estimate their sizes. And then the width of this transect can be determined from this. We're flying at a pretty consistent height above the seafloor, and the width of the transect can be measured, and then we get an area of the survey, of our survey effort, and can calculate densities of these various organisms, which make it possible to compare their abundance with other areas. You have to have the numbers per unit area sampled to have a comparable value. And also along the way on this transect, the ROV is equipped with the CTD sensor and other sensors that are measuring as we go along the depth, the oxygen, and the temperature. And so we have those environmental variables being measured along the way. And in addition, we're collecting the navigation data for this transect. So we have the exact location, and all of this information is connected by a timestamp. So it allows us, after the video has been analyzed and the data has been entered, it allows us to locate each of these animals along the transect based on their time. And then we also connect the location of each of these animals with a temperature value, a depth value, and the habitat that's being quantified. So we get really nicely detailed information from these quantitative transects. I'm going to change, I'm going to mute for a minute while I type in this change in habitat type.
Yes, covered by a little bit of fine silt and mud. This type of habitat, when it's been mapped using sonar, still shows up as hard. The sonar cannot distinguish between this fine layer of mud on the surface and bare rock. So what's the speed that we're at? Is this about half a knot?
Roger that, half a knot.
Thank you.
But it's definitely steeper than 18 degrees here. It's more like 45 there.
Yeah.
Okay.
Probably varying over it.
I'm adjusting the camera depending on the angle of the hill there to kind of get the same overall view.
So we're seeing some corals, some sponges, mostly sponges, different shapes, the vase shape. I think we just passed a heterocony. They look like waffles, shaped as in a waffle cone.
Mm-hmm.
First, E1.
Back to soft sediment, and you can see how quickly things change.
There's a heterocony right there. Yes, and I'm seeing the same gaping clams. Dan, do you know what kind those are?
Hope you're not asking me.
No, sorry. But you've picked up a lot, Dan, so it could have been you too. No, I was asking Dan through the science chat. He's from the Santa Barbara Museum of Natural History. I should have been more clear.
It's okay.
You all must have picked up a lot on the stuff that we're, yelling out just through osmosis.
Yeah.
There's a Dover sole.
Yeah. Yeah, Dover.
I'm passing the test.
Add one more step, 200 meters, bearing 040.
I just asked Tom to confirm
Thank you.
that we're seeing Dover because I started to second guess myself and wonder if some of these were deep sea sole, but I don't think so. Well, the deep sea sole are rounder in shape for one thing, and then they do have quite a bit of patterning, and they can get really big. And you see them in a variety of habitats. I've seen one vertically on a vertical wall in high relief rock. So they're not just a soft sediment.
You're a little soft again, Di, going in and out.
Okay.
That's
Thanks.
better.
I'm holding the mic up now.
Really cool looking terrain here.
Yeah.
Yeah.
Fractured.
How'd they get all those rocks stacked up this deep?
That's a Dover sole
Oh, so
and a
I think
hagfish.
Tom went back and looked at the tape during the descent, and he confirmed that it was a swordfish at 1233 or 1933 UTC. So if you want to get a glimpse of it in the left upper corner, you have a little thrill.
Oh yeah, I have to look for it.
Yeah.
Cool rock there.
Yes.
Yeah, slabs, aren't they?
Yeah. This matches the backscatter. It's right here, which would be, we're seeing the bedrock and intermittently some sediment patches, but still.
Yeah, so doing this transect is exactly what Channel Islands research team wants. They wanted to ground truth what the backscatter was showing. So this is great.
And you had said that you wanted to ground truth some of the darker spots, but perhaps not on this section, because we do have, right at the end here, we've got a dark patch just northeast. And then we also have this dark patch kind of to the left here. So
I think the dark, I think the waypoint EC was going to be in sort of a soft sediment area. I think
that sounds
that's
good
why
then.
Lizzie picked that particular waypoint.
Sure, then we won't deviate. We'll go with the original plan.
Yeah. Here's a very dirty sponge.
Yeah, that's 200 meters.
Should have been end of transect there.
Roger that.
Okay, end of transect.
Yep.
Well done. I'll type that in and mute for a Okay.
So I was just told that the dirty sponge, the nickname, I think it's a better nickname, is to call it the Cheeto sponge because it looks fuzzy. I like that better.
But it's not orange.
I know, but
I think you mean Cheeto sponge for the heterotone. If I remember correctly.
Yeah,
okay, yeah. Oh yeah, you're right. The
Instagram page.
heterocone.
Yeah.
Yeah.
Which one is the boot sponge then?
I think that's still the shaggy dog, right?
Oh, the orange heterocone, not the dirty sponge. I misspoke.
I think the dirty sponge is still the shaggy dog so far. I don't know.
So do the different watches on the ship get together and sort of share with each other, or are you all just ships passing each other in the night?
Oh, we hang out a little bit. There's at least two of the watches get together for meals, and then oftentimes for the ROV crew at least, there's either the offgoing or oncoming watch hangs out and helps to turn the vehicles around and get them ready for the next dive.
Okay.
So we often recover or dive at watch change. So we're sticking around 2 to 4 hours after our watch usually.
Yeah.
Especially if there's some more intensive maintenance to do.
Those are long shifts that you guys have.
Yeah.
Yeah, basically we work 2 8-hour shifts. That's how we roll on Nautilus.
Okay. Wow. Are you able to exercise at all on the ship? Do you have any equipment or walk the deck?
There's a little gym.
Yeah, small gym. Some weights, some machines, rower.
I heard somebody was giving a class.
That might have
yoga
been
class or something.
That might have been
like
Marley. One of the other video engineers, Marley.
And what is the class that she offers?
Well, I think it's very informal, and it's just people joining her at her workout if they want.
This is really interesting. I like this habitat.
Yeah, it's incredible the amount of bedrock, and I don't think we've seen coral yet, have we? Or at least it's not abundant.
Seen some mushroom corals.
Mushroom corals, yeah.
Oh, thanks.
Is that a clam in the lower left we just went over? I should call it out sooner so people have time to see it.
Yes, and Dan from the Natural History Museum said that they are a cesta, if they're sort of the gaping clams. There's a Pharea that we just passed a little patch
it's
of
on
it.
Is it on a heterocone that looks dead, maybe? And Tom says that these dirty sponges are the boot sponges.
Ah, thank you. Oops, sorry, wrong button.
So what is the distance between EA and EB?
Sure.
Approximately.
Just check that for you.
Interesting yellow sponge. Can we zoom in on that maybe?
Sure. You want to stop the
wandering sponge? Sorry to interrupt.
That's what we're here for.
Oh, and Abby just came on.
Hold position. Thank you. It's 800 meters there, Liz. Or sorry, Lisa.
Okay, Steve.
Cali.
800 meters. Okay.
And we have approximately gone 300, but we're doing a bit of a jog over. Can't quite recall if it was if you were on when we talked about it. But going up.
Oh, that's okay.
Jogging over.
I just wanted to.
Going up again.
We're staying in the white portion of the backscatter for a bit. We're zigging ever so slightly on our way to the next waypoint.
Good idea, and I think that that's what they want us to ground truth, where the hard is really hard. We're covered with.
So Abby says there's so many growth forms of this particular sponge, which they're calling a Macali, a yellow Macali. Macali. So there doesn't seem to be an indication of collecting, but just wanted to zoom in to.
It's a cool shape.
It seems to be an unusual shape, doesn't it?
It is unusual.
Yeah.
It's like a whole bunch of little ones have joined together. They're
like a chain, she says. Yeah.
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 And that some of them might still grow into each other, like that one piece that was sort of by itself, but maybe over a couple of years.
Yeah, you could sort of see multiple bases, it looks like. Yeah. Look at that.
It looks like Roman aqueducts.
Oh, yeah.
Zooming in.
Abby's happy. She likes this shot. Cool. Little shrimp. Isn't that amazing? So Abby, I have a question. Would you call this one single sponge or multiple sponges that have grown together? I have no idea. We'll see if she responds on the chat.
You see a little shrimp living there. So this is a great example of sponges and corals providing habitat, biogenic habitat. That's habitat that's created by a living organism. So it's becomes a place for other organisms to live their life, grow, reproduce, feed, do all the things that animals need to do to survive.
She's describing it as it has lots of little attachment points. So I think she's considering this one sponge. And this isn't a coral over here to the right, is it? It's a
Hydroid?
my mind's going blank. A hydroid, probably.
I just threw that out there, Di. I'm not sure.
No, well, I think you're right.
And then you can see lots of brittle stars here. At these depths, we see a lot of different kinds of echinoderms, and this is a group of organisms that have tube feet. So think about sea stars, sea cucumbers, urchins, and in addition to having these tube feet, they also have this radial symmetry in 5 parts. So as you can see here, 5 arms.
So Abby's saying you can see the osculae along the top of this sponge, and the exhalant water comes out of those larger openings after the sponge has extracted oxygen and food. Which, by the way, we're at pretty low oxygen saturation here. It's 0.25%.
Oh yeah, only 1.1 micromole per liter. Whoa, that is beyond the oxygen minimum zone.
And where do you see that value? I see the percent saturation. Where do you see that other, is there more in the quick stats window?
It's further to the right on the quick stats. So it's Herc depth, oxygen saturation.
See.
Yeah.
Yeah, I
and
just
O2
have to scroll
concentration.
down.
Yeah. I mean, each person has their Grafana set up differently, so that's how it shows up for me.
Yeah, I've got it now. Abby says she would call each clump of sponge an individual, but sponges that are growing next to each other could potentially be clones. That's really
so it just sort of, I guess, budded off and started a new colony that way. Yeah.
Oh, visitor.
Okay.
Hello. Oh, stop right there so we can see your third dorsal spine. You want to give the lesson, Di? Please.
Now I'm wondering if this is a short spine, because the third spine is not particularly pronounced on this one. So this might be a short spine thorny head rather than a long spine thorny head. But we are getting
sweet.
a good view.
It just landed right where the lasers were.
That was
very
perfect.
nice.
It's a quick measurement.
Demonstration.
And cut.
My peach, yeah.
Now if we could have the next contestant land right in front of us.
And Tom says yes, it is a short spine. Yes.
Yeah. Oh, good. Got that one right.
Tom confirmed.
Okay. You guys ready to move?
All right. We can. Yeah, we can carry on. Thank you.
It was beautiful.
Abby adds that many sponges are hermaphrodites and can reproduce sexually, producing larvae that swim or crawl, but some can also reproduce by budding.
Ooh, this is a pretty vase shaped one.
So is that a Cheeto right there?
Yep.
Okay.
I think so.
Looks like a
You were
bugle.
confirming that,
yeah.
Taylor Ann? That was a Cheeto?
So, yeah, the heterocone. If you look on their Instagram page, I think that they posted that a scientist or someone was hungry and called it the Cheeto.
See the white one or the yellow one?
The yellow, or yeah, the yellow one over there with the, I guess, I don't know.
But the white one's pretty too.
Yeah.
The white one's pretty too.
Yeah. With the crab going into defense posture.
No food reference. This is beautiful habitat for sponges. And then in the background of the Cheeto sponge, we can see the dirty boot sponge, I think is what Tom called it, or the shaggy dog. We have all kinds of names for these.
So this heterocony that is the heterocony here, beautiful vase sponge on the left, is the Cheeto sponge. Is that what? Got to get my
yeah,
the unofficial
colloquial name.
Yeah.
Yeah.
Okay.
Get your
now that makes more
down.
sense. That makes more sense to me because it's more orange.
Oh, there's a great view from the Herc cam. Folks want to watch channel 2 or the quad view on channel 4. You see the different sponges that we're viewing.
It's like Dr. Seuss instruments.
What's that?
These look like Dr. Seuss instruments.
Oh, yeah.
Yes, I agree.
Yeah. Every once in a while, be sure to look at channel 2 and the downward view from Argus, the second ROV, looking down upon Hercules. I like that view a lot. It reminds you of how dark it is down here and how we're kind of searching along, going along with the flashlight compared to...
Basically, yeah.
It's a pretty cool angle, Dan. Do you want to kill the lasers for a second, Jake?
Sure.
Yeah, nice shot.
Yeah, very nice. Beautiful. There's some chat going on in the science chat about what eats sponges. Sea stars do, sea slugs also eat sponges.
See
and
all
Abby
the
says, okay.
The shrimp and the smaller inverts just drifting by. And it seems as if some of them, as they drift by the sponge, they're trying to hold on like, stop.
You zoom in there, Steve, you'll get a full screen sponge.
That looks like a brachiopod over to the left on the bottom as well. Oh, look at that detail. Wow.
So those pores are lined by, I want to say cilia, but they move the water from outside to the inside of the sponge. And then that is the outgoing water.
Are those, what are those, egg sacs or something in there? Those look like the yellow things that we thought were sponges the other day.
Might just be the backlighting, unless I'm seeing something else.
Yeah, I think that's just seeing the light on the seafloor through the sponge.
Oh, really?
Yeah.
Wow.
Mm-hmm.
Okay. Interesting. Yeah, you're right.
It's like an optical illusion.
Yeah, you could look at it as like protruding towards you,
Yeah.
but it's inset. Yeah.
So weird. Wow, that's cool. What detail that
Amazing
camera
architecture.
Has.
Yeah.
So you might, from these shots, think that sponges are like the sponges at home that we use to wash our dishes, but they have a skeleton made up of glass spicules, so they're actually quite crunchy, not soft and fuzzy.
Kind of an interesting lobe off the top hanging down. It looks like it's part of the sponge, but it's different than the rest of the structure we're seeing. Abby's loving these shots.
Great, beautiful.
Thank you. Amazing.
Well, and because you could see as we were looking through the windows here, you can see that the background is green, and so greenish gray, which is what the substrate color is. It's like these little windows with gauze.
Stained glass.
Yeah, that's much prettier description.
That's a screensaver, isn't it? This shot. Lovely.
Where's the, oh, I'm too close for the DSC helmet.
Yeah,
Oh well.
just over it.
As we come out, I thought I saw a brachiopod on the bottom to the left as we zoom back out when we're ready. And Daniel was interested in that. If indeed it is a brachiopod, he missed it when I mentioned it. So
Was it at the base? We're just on
we'd have to zoom out more, but we don't have to right away. Where did I see?
You gotta hold each one for, what's the rule, 10 seconds or 30 seconds? Or?
Well,
now I'm not sure I'm seeing it, so my eyes may have been playing tricks on me.
Is it the white?
Well, he did in the science chat say.
Well, he said he missed it, I think, in response to my
zoom in on the base
comment.
there, though. That'd be a good shot of the bottom.
I'm wondering about that rock to the left of the sponge in front of the thorny head.
do you need me to zoom out?
Oh, I didn't even see that sponge down there.
Boot sponge.
The boot
Yeah.
sponge. Blends right in.
Cool.
Yeah, it totally disappears in the background. The brittle stars know it's there.
Wow. Yeah, they sure do.
Is it actually growing on another sponge?
It looks like it. Yeah, I think so.
What types of stuff going on?
That one brittle star is like buried in that sponge.
Oh yeah.
Abby says we are interested in these because the morphology of heterocony calyx is so variable. These look very different from the ones off Washington.
Yeah, they do.
She's not asking for a sample, but Abby, chime in if that is coming to mind.
That is interesting, Jake. That one that looks buried.
Yeah, no, it's like under all the.
Yes, it's encased.
Help me.
Yeah.
I wonder if it's stuck.
Yeah. Yeah, like
look at those
Harrison Ford.
It's not just science fiction.
No.
And then is it even on top of an encrusting sponge of some type, this white at the base?
What's that pink bit down there?
Is it a little mushroom coral? I can't tell.
Or a little.
You have to zoom on it.
Rotate there if you want.
I think they're looking at that pink in the bottom
mushroom
right.
Coral.
Yeah.
Baby.
Yeah, what do you got down under there? Crab.
Yeah.
Oh,
Crab arm.
There's a crab.
That is a home if I've ever seen one.
Excavation there. Look at that.
And Tom was saying that pink organism or blob to the lower right, he thinks it's a small mushroom coral, like what you were saying, Taylor Ann.
Seems to have maybe one big polyp out, the rest are closed. Couldn't quite make out what was happening there, but maybe that's what it is.
Oh yeah.
Polyps just seem large for its
Yeah,
size.
it looks retracted.
Oh, cup coral.
Is that a sparkling?
Yeah, cup coral, mushroom coral.
Go tight there, Steve.
Full zoom.
Man,
Maybe a feather duster worm.
you really see
in
so
there
much
too.
when you zoom.
Yeah.
Yeah.
Right. Maybe.
Yeah, so we don't need to collect here. They
No
collected
sample. Yeah.
a sample yesterday, so we're good.
Zoom in on the.
Thanks, Abby.
It's full zoom.
So those are all sponge spicules on the dirty one, not something else also growing on it.
Looks like there might be hydroids on there.
Yeah, I
That's what I
agree,
was
Taylor Ann.
wondering. And then who are these little patches of white dotted something or other?
Maybe an encrusting tunicate or...
Yeah, it looks like maybe a tunica.
Abby might know.
Bryozoan maybe. Yeah.
Bryozoans maybe. And the crab looks like Corilla, he says.
Okay, I can zoom out now.
Corilla davila.
It's an impressive ID since we couldn't hardly see it.
Yeah.
I know.
Tom
like,
Tom
how
can do
can
that.
you tell?
And he can do it with some authority too.
Yeah.
I'm convinced
Yeah.
Tom has a magnifying glasses for eyes.
And there's a hagfish coming in to investigate what's going on here.
Yep.
They have those eerie, shiny eyes when light shines on them. They do truly look like the Walking Dead or the Slithering Dead.
They always show up
They
unannounced.
They produce a lot of slime if you catch them. But if you have an eel skin wallet, it is likely made out of hagfish skin.
Yeah. It's a quite profitable fishery off of our coast. I think it's the third most profitable fishery for the Morro Bay Harbor.
Oh, really?
Yeah, one of their clients are come from Korea, quite an export there.
How do they fish for hagfish?
I was just about to ask that.
What's that?
I think they do traps, don't
I was, well,
yeah,
I, was,
I think if
I would say
they
that
bait a trap.
on ONC we
they're
put
kind of
down
they're
crab
a
traps
scavenger.
before, and all we catch is hagfish. So
yeah.
apparently they get caught in those crab traps.
Yep. So traps. And I was reading that not only for their skin. Oh, what is that drifting by? It's a type of eel pout?
Oh, I don't see it. Oh, over to the right. My video.